Yeah, now, Dr.
Okay.
Sinha, now you can forward. You can start your session. Yeah.
Thank you, Dr. Thomas. That was a really modest and very nice introduction, and I'm really overwhelmed now. And
Thank you.
I really want to thank Dr. Supriyo because he arranged all this. Because he was telling me in the beginning of January that he wants to hold one webinar, and so yeah, so I'm really full. I mean, so much thank you to him. And I really welcome along with you, Dr. Thomas, all the VC sir and other faculty members and students. So let me share my screen first, and then I can move. Okay. So, while I was talking with Dr. Sen that how we are going to do this session, so I wanted to, share what research that we do in our company and how we are involving different streams of science, engineering also, to develop products. So I will give you that overview so that students will get a feel of what kind of products we are developing, and not just that, the science behind that, because that is very important at your stage of studies, that you should understand what is the science behind everything, behind research, behind if you are developing an innovative idea. So just understand that science. So that is what I will be starting this, what we do, and then I will move towards the other modules and what other things that we do in our company, which might be very beneficial for you and could serve as a starting point in your next career endeavors. So I'll be telling you. So basically, my company is named as BioAptagen, right? So I don't know if you have heard the word aptamers, uh, but so aptamers is kind of a Greek word, right, which is called aptus, which is to something that fits. So my company basically makes, I'll be telling you in detail. So our company basically deals with, we generate DNA aptamers, which we use them for our diagnostic purposes. So in the past, when you'll be listening about or reading about different techniques like ELISA and antigen antibody based interactions, so most of the diagnostic kits or most of the diagnosis that takes place, it is basically protein based, right? But it has its own upsides, it has its own cons, pros and cons. So in the past, probably 10 years, DNA molecules and RNA molecules, they have come up as a very handy, very portable, and highly sensitive also precision tools for diagnosing various kinds of human ailments. So they are soon replacing protein molecules or the antibody molecules to emerge as a next generation diagnostic tools. I'll be talking about that. So let's first understand in a very, very simplified manner that what are these biosensors or electrochemical biosensors are, right? So when you go to a doctor, right, and if the doctor recommends that, if you say I have some pain, I have some kind of symptoms, so doctor recommends that you go for some diagnostic test. He tells you that go on a diagnostic clinic and just get your blood tested or anything. So what really is tested in your blood or in your urine or in your cerebrospinal fluids? Like these are the three main biological fluids. So there is some biomarker always, right? It could be a protein biomarker, it could be a fat molecule, it could be vitamin, all the biomolecules. But then these biomarkers, how are you going to detect it? It should be present in your system, in any of your biological fluids, so that it could be detected by some other molecule, which will serve as kind of a detector molecule, right? And then it should give some kind of a signal, because these biomolecules are very, very small in size. Okay, so to get a signal, they should show their presence, that they are there in your biological fluids. Okay, so that signal could be in the form of light, it could be in a form of fluorescence, it could be in a form of color. These are the three main, or some kind of electrical signal. Then these signals could be, you could see them by your naked eye if there's a color change, or if there's a fluorescence, you could read it on a UV, any kind of signal. And then there are two levels. Once when you see it, it becomes, the test becomes qualitative, that you know that it is there because you are seeing a color. But then, just to be more precise, these signals could be converted into some kind of an electrical signals, right? And then they're processed, and then you get a quantitative reading. So you might have seen your parents or family members using those glucometers that are seriously popular everywhere. When you get this kind of a reading in which they have a digital little screen, and then you get the glucose amount that is present in your blood. So basically, there's a biomarker there, glucose or any other HPCTs. There are many other molecules, and then those are the biomolecules, and then you have a antigen or antibody, whatever. So it binds to your biomarker, it gives some kind of a color, and that color is then transferred or transduced into some electrical signals, processed, and then you get a reading out of it. So this is how any kind of a sensor works. But then if this sensor is, you are doing it with some kind of a living molecule, a living individual, so then it becomes a biosensor. Okay. So why these biosensors? Why you use these biosensors? Because see, there are many, many plus points for that. They are highly sensitive. The response is really fast. When you test blood sugar or any kind of prick test, the response time is very fast, right? Like in within 30 seconds or within 1 minute, you get the result. Also, they're very simple in operation, which is very handy because you can use it at home. It could be used as a point of care device. And most of the time, it has been, scientists think that it should be very much cost effective, right? Because there are a lot of population who cannot afford these biosense, any kind of diagnostic devices. So there's always an aim that whatever devices anybody's making, it should be low in cost, right? And then of course, it should be very point of care. So you must have realized the importance of point of care during the COVID pandemic, because then you have to go for the whole exercise for the RT-PCR testing. It takes 3-4 days for the results to come, and within that time, the patients, if the patient is really COVID positive, his symptoms might pile up, right? So all these things can be avoided. It could be the response time. The first responders could be very fast if your detection is fast and very robust. Okay? So let's see, when you talk about DNA apta sensors or DNA sensors for that matter, what is the DNA in that sensor, right? What is that DNA molecule that we use in making those apta sensors that we make in our company? So like I said, there's a molecule called aptamers, right? So basically aptamer came from a Latin word, like most of the words in science, which means aptus, which means to fit, okay? And meros is a region, whatever. So it becomes aptamer. So this is a DNA molecule which specifically binds to its ligand, identifies its ligand. So if this DNA molecule is specific for, say, your glucose in the blood, so it will only and only bind to your glucose molecule and none of the other molecules. So you will ask a question that what is plus or what is great about this DNA aptamers that is not there in antibodies? The answer is DNA molecules, they're the most robust molecules found in nature. They're very robust, they're very inert molecules, right? They're very thermostable. And protein molecule, antibody molecules for that matter, they are mostly for the proteins, protein antigens. But DNA molecules or DNA aptamers, you can develop these aptamers for anything. Basically, these molecules are single-stranded, and why they become why the molecular recognition is very, very specific. That is the reason, because these DNA molecules are single-stranded molecules. They're synthetic molecules. They are not found in nature. They are single-stranded molecules, and when any nucleic acid molecule is single-stranded, it is they form a folding structure, and it gives that DNA aptamer molecule a specific conformation. Okay, and this folding structure happens because all these nitrogenous bases, they enter into hydrogen bonds. They are of course not covalent bonds, but they enter into hydrogen bonding, and that gives it a unique conformation which is sequence specific, right? And any two DNA aptamer molecules, they are not of same sequence. So any two DNA aptamer molecules, so there's a very wide diverse conformations of, and that is what it provides these DNA aptamer molecules a specific molecular recognition quality. Okay, these are two examples. So this conformation, say for example, this is specific for one specific kind of a ligand. The same way as antibody, you must have read in your immunology classes, those VDJ gene displacement happens, and which gives rise to an antibody unique molecule specific for an antigen, and then clonal recognition happens. So just keep that in your mind. So I'll tell you how we develop this DNA aptamer molecules, and then you will form a stark analogy between how this VDJ recombination takes place in your body for making the unique antibody molecules for specific antigen, and it is actually replicated each and every step in vitro on the lab bench, and that's how we develop these DNA aptamer molecules, which are then specific for ligands. So, like I said, that DNA aptamers, they are more diverse and versatile charismatic molecules as compared to protein, because in the case of antibodies, your ligands are mostly they are antigen molecules, they are proteins. For DNA aptamer, there is no such thing. It could be as diverse as metal ions, organic dyes. The ligands could be amino acid, proteins, even the whole mammalian cells, viruses, full viruses, and bacteria. So like in our case, for our center stage product, that is the UTI detection kit that we are developing. So in this case, the DNA aptamer molecules, they bind to or they identify specific bacteria, right? So they're versatile molecules. It could bind to any given, even the another DNA molecule also, right? So that is a diversity that these aptamer molecules provide. So what we do is we in our company, we actually combine molecular biology, synthetic biology, and nanotechnology for developing affordable diagnostics for different healthcare products. So in this meeting, I have with me my team members also. So Sahil is there in my team. So he is basically, he has another company in IT campus, which is called as Primary Healthtech. I'll be telling you about that. So he is the guy who actually, he's a nanotechnologist. And so what he does is we make DNA aptamers, and then he converts those DNA aptamer molecules into gold nanoparticles, and then whatever signals, color signal that we get, again, like I was telling you in the beginning, he converts those color signal into electrical signals, and then we get a quantitative readout. So right now, what we are doing in our company is we are developing UTI detection kit, which is almost ready for clinical trials. We'll be putting it in for clinical trials in like maybe a couple of months after. And then we have conceptualized a cardiac malfunction kit, with which we have an aptamers which are specific to a cardiac function biomarker, which is BT pro and anti-BNP in the blood. And then we also started work for vitamin D3 detection kit. Again, this is apta sensor kit. And plus, we are also doing a one COVID detection kit. I'll be telling you in more detail that it's not just an apta sensor kit that we're doing for COVID detection, and we're actually preparing a model system which could detect any kind of viral disease, not just COVID. So like I was telling you, how these aptamers are made, because then you might have in your mind all the the biological processes that takes place in your cells when the antibody production happens. So you must have read of those VDJ recombinations, and then a lot of antibodies are produced, and when there is an antigen, then all these antibody molecules, they just pile up around that antigen, and then clonal recognition happens, right? So that particular antibody which binds specifically to that antigen molecule, then that antibody is selected, and then it is enriched, which is called as clonal expansion. The same thing we do in the lab for selecting and for making these aptamer molecules, right? So there's a process called systemic evolution of ligands by exponential enrichment, which is acronymed as SELECTS. So this is the method that is used in the lab to develop and evolve these DNA aptamer molecules for a specific ligand. Okay. So it might look a bit complicated to you, but believe me, it's really easy to understand. So just imagine we take a library of DNA molecule, right? And this library has around 10 to the power 15 DNA molecules, that a huge number of DNA molecules. All these library members are of same they contain DNA aptamer molecules of same length. So let's say what the library that we used in our lab, it is 70 bases long library. Okay. So we have a huge library which has 10 to the power 15 DNA, unique DNA members, so one molecule for each, and it is 70 bases long. And then if you see this figure, it has 5' end and 3' end, somewhere around 15 base pair bases of these ends of these aptamer. They are known to us. Why? Because I'll tell you afterwards why we know. So this mid part, this 40-mer part, this is randomized. We don't know the sequence. So that is unique about one particular DNA aptamer molecule. So what we do is we take this library and we incubate it with the ligand for which we want to develop this aptamer or evolve this aptamer. This ligand could be anything. It could be a cell, it could be a virus particle, it could be a metal ion, it could be a protein, anything you want, any biomarker, whatever. So what we do is we incubate it and then let these two ligand and this library members interact, right? So what will happen? Few DNA molecules which have some kind of a conformation, they will attach to this ligand cell molecule, whatever, and rest of the things will remain unbounded. So then we do subsequent washes. So we collect only those DNA aptamer molecules which specifically bind to our ligand. Rest of the things, they are washed off. Now DNA molecule, I told you this entire library, there is a single DNA molecule, a single aptamer molecule for each the diverse library member. So, and you cannot just take out one small 70 base DNA molecule. You just can't, right? So what you do a PCR. So PCR, what it does, it will amplify or enrich those DNA molecules which are attached or have been chosen for this ligand to bind specifically. And then that is how this design will come into picture. So when you do PCR, you need a DNA primer. Okay, these DNA primer, they attach to this part, 5' end and 3' end. So two primers, forward and reverse primer, they will attach to these two ends, whose sequence I told you is known to us. Okay, so we design primers against this 5' part and 3' part. And with these primers, we will amplify or enrich those DNA aptamer molecules which are uniquely identifying your ligands. Okay, so once we amplify it, again, when you do a PCR, the DNA that you get, it becomes double-stranded. Okay, but for these aptamer molecules, they need to come into that unique conformation to bind to this ligand, right? So we have to make these double-stranded DNA molecules into single-stranded DNA molecules. So we do that part, we do the PCR, then we make these DNA molecules single-stranded, and then we again incubate it with the same ligand. So this entire cycle, we do this iteration rounds of binding and rebinding and PCR amplification, making it single-stranded, again binding. We do this iteration round for around 10 to 12 cycles. Why we do it? Because we want to be sure that whatever DNA aptamers we are getting, they should be uniquely identifying your ligand molecules. So after we are done with 10 or 12 rounds, we are like 99% sure that whatever molecule we are getting, they're specifically identifying our ligand. So then we enter into the second step. So remember I told you the DNA aptamer sequence is unknown to us, that 40-mer sequence between the two primal limits. So now we need to know the sequence. So we go for NGS platforms or next generation sequencing platforms. We generally do it with Illumina on Illumina platform, right? So we do the NGS methods, and then we know these are DNA aptamer sequences which are uniquely identifying our ligand. So this is just an output of one of our DNA aptamer library, selected library molecules. So here, the sequences that we get from NGS, they are unique sequences of about 200 or 500 unique DNA aptamers. So remember, we started from 10 to the power 15 molecules, and now we came down to 200 to 500 unique DNA aptamer molecules. Now we cannot test these 500 individual molecule for your DNA, for our ligand, right? It will be logistically impossible. So there comes our third step, in which we do aptamer candidate optimization. We do that by using AI or artificial intelligence and machine learning algorithms. which actually we put all these sequence information inside the in these algorithm, and then it just gives you some hits which of those aptamers which will be the best binders to your ligand, and then that 500 number comes down to 10 to 20 candidate DNA aptamers, right? So now 10 or 20 candidate DNA aptamers, you can easily do test these individually for your ligand experimentally. So what we do is, once we are down to 20 candidate DNA aptamers, we synthesize these DNA aptamer molecules from the oligosynthesis companies, and then we do experimental testing of each and every DNA aptamer molecule for all these parameters like binding specificity, binding affinity, structural stability, all these things we do, and then we come up with somewhere around 2 to 3 best binding DNA aptamers. So we started from 10 to the power 15 molecules, and we came down to 2 to 3 basically the best DNA binding aptamer candidates, uniquely and specifically identify your ligand for which we are developing. So these are the various steps that we perform routinely in our lab. And after that, so these all things, they are done by us in BioAptagen. And after that, like I said, Sahil takes over, and then what he does is he attaches those 2 to 3 best candidates to the gold nanoparticles. And here, these aptamer molecules are converted into apta sensors, right? So these DNA aptamer molecules, so he has different methods for attaching these DNA aptamer molecules to gold nanoparticles. If somebody wants to ask, he can ask them later on. So once this DNA aptamers are converted into apta sensor or nanosensors, now they are ready to detect your biomarker in any biological fluid. Okay. So and what happens is these gold nanoparticles, they have a unique wine red color, and once they attach to the so this is how the test goes. So say, for example, this is your unique DNA aptamer molecule, and you have converted them into a gold nanosensor or apta sensor, right? So what happens is, as soon as this nanosensor gets a target, so these gold nanoparticle, they just, these aptamer, they will bind to this ligand molecule. In this case, for example, any virus or any bacterial cell. So there is a color change due to the aggregations. So, and this color change is visible color change. You can detect that, and or this color change could be converted into a electrical signal, and then you can get a a quantitative reading. So that is what we are doing, and this we are doing with, like I said, with Primary Healthtech and with IIT Guwahati, is that we are developing these aptamers into apta sensor or nanosensors. Okay. So there are other things that we are also doing in our company or developing products. Like I said in the beginning, we are actually developing a lab PCR based viral detection platform. So I don't know if you must have heard about LAMP PCR. This is ligation amplification. This is kind of a complex PCR method in which you have 6 set of primers for a single template, and then they amplify your DNA template molecules in a very dumbbell kind of a loop. This is kind of a loop mediated PCR, right? So whatever product you are getting, it's a looped DNA kind of a thing. And after the PCR is done, you get huge amount of DNA. And that huge amount of DNA, it changes the pH of your reaction mix. And that pH, we put a pH indicator, and there's a change in color. So like in the beginning, we have a dye we put in the reaction mix. It has a different color because the pH of your solution is different. Say, for example, generally we set the pH at around 8 or something. But when the PCR is done, because there is a huge amount of DNA that is formed by lab PCRs, so what will happen is, because DNA has a negative charge because of the phosphate backbone in the molecule, that changes the pH of your solution, and the pH goes down to around 6. And that is enough for that pH indicator dye to change the color. So we get a color change. And the idea of developing this lab PCR based detection kit is it can just curb the use of RT-PCRs. It could be made point of care because, I mean, RT-PCR, they're expensive, and it cannot be made point of care. You have to give the samples to the, to the diagnostic center, and they take their own time, 2-3 days to come up with your final result. Here, you don't have to do all these things. And this PCR, it doesn't need a PCR machine, because there is no cycling procedure here. You just put the entire reaction mix at around 63 degrees centigrade and let the reaction happen for 40 minutes. And after 40 minutes, your loop DNA is formed, which changes the color of your solution if it is there. So we're developing this platform for any kind of viral detection. There's a different kind of polymerase that we use here, not the regular Taq polymerase. We use BST polymerase. And so what Sahil is doing, so we are actually making those buffers, and we are trying different kind of indicator dyes, and then Sahil will take over, and then he will convert that color signal or change in turbidity or anything into some kind of a quantitative signal. So that is his part he'll be doing. And another that I already told you, we are actually developing after-sensor-based vitamin D estimation kit in serum. And also we have started R&D of a NASPA method, which is also used for viral and bacterial detection. So this, all these, so next generation, like I told you, the next generation of diagnostics is all about molecular diagnostics, where actually you are detecting directly the DNA or the RNA of anything, bacterial infection, viral infection, anything of that, maybe fungal infection also. So NASPA is another kind of isothermal PCR method, like the LAMP PCR. Isothermal means you don't do cycling, because when you do PCR, you have this denaturation step, then you have amplification step. So there are 3 different temperature settings in that. Here in all these isothermal PCR, you use just 1 temperature. So you don't need a big PCR machine for that. You can just take a heating block, which could set the temperature at around 60 or 42 degrees centigrade. Like NASPA takes place at 42 degrees centigrade. And there's a set of enzymes, and just set the temperature at one number, and then you can get the entire results in say about 40 to 50 minutes, right? So that is what this is under a very initial R&D stage, but we hope that it will come maybe in next year. So besides doing a lot of R&D work, like Dr. Supriya pointed out in the beginning, we also do services. I'll be detailing that part in a while from now. So that we call it as a contract research services, which is an RE-genome, we named it. And then one which might be very, very useful to all the students who are attending this webinar, we do educational, we are hosting an educational program for the students where you get unique hands-on laboratory experience, and we have named it as a Nurture Youth Program. I'll be telling you detail about that. And then, during the lockdown period, we also formed a hub with 3 other entrepreneurs, and we have named it as Wannabes Hub, where we provide mentorship support for developing any idea, entrepreneurial idea, into how to convert that ideation to a venture, right? So if you have an innovative idea, how really you are going to graduate from ideation phase to startup phase, and then how you're going to raise funds, and how you're going to even write grants for different funding agencies, be it R&D grant or a corporate grant, anything. So we have this forum, and you can just go on the website and find it out more about it. So now the second part of my webinar that is actually might be very, very beneficial to the students who are attending it, that all is focused on how you're going to choose your career after you finish your degree, right? So most of the time, you ask your parents and what your friends do, what your seniors have done, right? And you just follow the path, right? But most of the time, students don't think that what actually they love doing. Okay, so if you have chosen science as your study stream, then you should also know that what are you going to do with that education of science that you are doing or getting that degree since 3-4 years. So you have to put that not just on your parents' advice or not on your friends' advice. I've taken science so that I can get grades, I can apply for this job, or maybe I could get married to a good guy, all those things. So but then back of the mind, there always be or should be in front of your mind that why you have chosen science, right? So, and what career option you have, and which could actually help your scientific education that you have gained, how you're going to apply that. Do you really love doing that, right? So you have to ask this question, and it is not just about going into that sweet spot that it is going to pay me well, or because for some extent, for some years, you will like everything, but then it becomes monotonous. So if you don't love what you're doing, then you lack that passion afterwards, So I always suggest students, just come out of your comfort zone and take a leap of faith, because don't try to be a part of a crowd. And right, you can, if you have a capability to go away from the crowd and shine on your own, just go for it. Okay. And that is how the three golden rules that I describe for every student who come in contact with me, that choose your career option which actually you love doing. So if you have chosen science, go beyond your degree in that arena of science which you love doing it. If you really love research, then go into research. If you love industry jobs, if you want to develop a product, just go for that. Okay. And then if you know what you love doing, then you should train yourself in the skills that are needed to pursue what you want to do. Okay. And that is where we come. Okay. So we help you in training yourself in the necessary skills that are needed to define your, or to be in that career endeavor that you like. Okay. That is what we do. We fine tune your science experimental skill, because all the theoretical know-how you are taking from your eminent faculties through, you're pursuing your degree at whatever level you are. So you go your classes, so all your faculty members, they actually tell you, teach you, right? You gain knowledge from them. But then what we do is we fine tune your experimental skill. Okay? Theoretical part, we know that your teachers teach you in the best possible way. So what we offer you uniquely designed lab training modules, right, in which you are going to set up your experiments by your own hands. And there will be no peeping, right, you'll be doing everything on your own. Basically, we will be telling you how to become a researcher. This is all we can say, like in the army, there is one NDA, is a National Defense Academy, and then there is IMA, or Indian Military Academy. So in NDA, they actually tell you what is a soldier, what is being in army. In IMA, they actually put you in the arena, in the war zone, and they just let you. So that's the experimental part. So this is, we can say, this is a field study we do. Whatever theory you are gaining or still gaining from your teachers in your back in your universities, what we tune in experimental part, what you actually do on the lab bench, right? So we have state of art and infrastructure support, and we are incubated in IIT Guwahati. So you have all sorts of infrastructure exposed to you, right? So we have different modules. We have modules from molecular biology, microbiology, nanotechnology, population genetics, industrial engineering, and there are many other fields. These are the main fields that I have listed here. So in all these fields, we provide you a hands-on training program, and not just that, we have a full design modules in which we tell you, like I'll just, for example, so we started this program in 2019, and I think few of your seniors have visited our lab, and I think 6 or 7 students have gained training from our the last season. And these are their pictures, like, if I'm not wrong. So right, so we started this program in 2019, and the idea behind this is only that because I did a little teaching sprint in Assam downtown university here, and there I realized that students are not that much exposed to the cutting edge infrastructure support that can fine tune their experimental skills. And that is how I realized that in Northeast lacks that part. So that is why, and being in IIT and being in a lab and hosting a lab where we have all sorts of state-of-art instruments, we will like the students like you to train yourself under those facilities. So this is kind of a joint initiative that is from BioAptagen and Primary Healthtech. So Sahil's company is Primary Healthtech, and they are also incubated in IIT Guwahati. So we have jointly made those many modules, and recently we also conceptualized another company. So we have started the procedures for making that company, which is called as Arcus BioSciences Biosolution. So that will be dedicating totally, completely to this training purposes. Right, so this is kind of just an example of what modules that we have right now in our company that we offer. So this is a 15-day internship program. So you can see that we have divided that. We have the DNA methods here, we have protein methods, then we have our microbiology lab methods, and we also tell you how you're going to troubleshoot your experiments, right? So it is not just, I mean, we give you the whole protocols and then we tell you how actually you perform your experiments. We handhold you, how you plan your experiments, how you multitask your experiments, how you're going to write your experimental data in your logbook, and how you're going to interpret your results, right? This is a 15-day internship course. This is the most popular one. And then we have a 1-month program. This is quite an extension of that 15-day program, but we have some kind of additional methods like cell biology methods. We have a fully functional cell biology lab, and then we have bioinformatics methods. We also teach methods like, that are commonly used that you will be, if you are going to pursue in research and want to do PhD and go beyond, you'll be needing all these methods, because most of the time I've seen students, even the PhD students, when they join a PhD program, they're completely lost. They're not, because they're not exposed to all these things. So we just tell you how to do that. And then we have a journal club where, because I think that lacks most in students, they don't know how to present their own data. Right? So we fine tune their communication skills also, where we tell them how you're going to present your data in front of others, and also how to write your data in your book, how you're going to interpret your data in different ways and results, right? And then we have some custom programs for specific techniques, like we have a total program dedicated to PCR proficiency, in which we tell you about different PCR methods. It's a very beautiful program. You'll be learning about endpoint PCR, that is the most easier one. And they'll be telling you about qPCR, how you're going to interpret your results, how you're going to design experiments for qPCR, then different other PCRs like lab PCRs, colony PCRs, diagnostic PCRs. So even the primer probe designing, we do all kinds of primer designing, how you design the probes, all these things. So if you want to apply for industry jobs, right? You will learn all these things, and then you will just make your CV stronger. And then, if you want to apply for different jobs in the industry who are actually working on PCR diagnostics, so that'll be very, very helpful for you. And then we have another program for the students who are actually working in the population genetics area and in genetic variation or the polymorphism studies that they do. So we have custom design programs for them, and this also anthropology students can participate in this program. We tell you about different kinds of DNA polymorphism experimental tools like RFLP, ARMS PCR, etc. We tell you about the pedigree analysis, how to interpret the results from next generation sequencing data, which is like a voluminous data, and how you annotate that data into gene spots, and all those things we'll be telling you about that. How you retrieve the different databases that is there, all the bioinformatics tools. So all these, you can go on the website also, it is there. Or I will be sharing you the contact details. You can directly contact with us. So these are the takeaways of your courses. We'll be giving you all the detailed protocols, theory. Generally, what we do is we take a theory class of what you're going to do, and we explain the method, what will you be performing, and then we let you perform it, right? So you will do it on your own. So we have different pipette sets for students, and they do it by themselves. And we'll be also telling you about biosafety guidelines that are very, very needed when you go, especially for industrial jobs afterwards, right? Because they have to comply to the different biosafety guidelines, and they expect that each and every employee should know these guidelines before they join their organization. And of course, all these programs are certificate programs. You'll be getting a certificate of whatever course you're doing. So plus that, we also provide dissertation projects, and if you, because I know in your curriculum, whether it is the bachelor program or your master's program, you have a mini project that you have to do as a part of your curriculum. So we provide that facility also. You can come to our lab and you can just do the entire project, dissertation project with us. So these are the current topics that now we are running. I think we are hosting 5 students from Assam Downtown University in like next week. So they are coming for dissertations. So they'll be doing their entire dissertation of 3 months with us. And these current topics, they change, because as soon as we keep on adding new ones, we could keep on upgrading from our old projects. So that we do. We can talk it over if you're interested, and we can talk it over, and then we can custom design our project for you. And so whatever we do here, so these are different salient features. You can go on the on our website and you can just get a better idea of what we do, and you can just directly talk to us. And when students do dissertation work with us, it's not just we tell them you are going to do this. We give them research articles and we enter it. We call it a brainstorming session where they tell this is not coming the way it should come, and what might have gone wrong. Or maybe sometimes, most of the time in science it happens once you are anticipating something but it is not coming, which is actually something new you have found, right? So all these brainstorming sessions, we do that with our students, and they find it really interesting because then that actually stimulates their brain, and you have all those adrenaline flowing, and then you get a lot of good inputs in your experiments by yourself. And these are the modules that are specifically designed for engineering students. So Sahil is there. So I don't know if we have any engineering students here, but you can pass on to your friends and to different faculties. So these are the modules that are actually designed by Sahil. So he has designed a spectroscopy module in which you have instrument introduction and how the theory part and the laboratory training, and then you have electrochemistry module. So all these things are there. You can ask him directly, or we'll be sharing his email ID also, so you can contact him or us. And then you can just spread it with your engineering counterparts. And then also you can have your own project, and suppose you don't have that kind of infrastructure in your universities, and so you can just join hands with us and we can do a combined project together, right? So if you have your project idea, and if you don't have that state-of-art instrumentation or anything, anything, so you can just come to us, you can do that also, right? So we call it like do your own project, and we'll provide you all the infrastructure support, equipment support, intellectual support, right? And also the brainstorming session, they're always there. Will help you in developing the publication quality images, right? So all these things will be helping you with. Eligibility is, any streams of life sciences. It could be biotechnology, microbiology, pure sciences also, any field of science, right? And also engineering. And even we are open to the students who are not currently pursuing any degree, but they want to, increase their skill for their CV. We also help them out. These are my team members, and they're actually all are present right now in the meeting. Sahil, I already told you. So Moshmi is one of my first employee, and she's kind of a backbone of our company. She joined in 2018 when we formed the company, and Reena has recently joined us. And so me and Reena, we are launching another company that I told you about a while ago, that is Arcus Bioscience and Biosolution. So that will be totally, completely dedicated to all these training purposes. Right. So right now she's a nursery youth program coordinator. And so we have advisors also, different advisors who give daily inputs on, business sense, entrepreneurial skills, and the scientific advisory committee. And this is our contract research services. So we provide different services. This is the catalog. We'll be sharing it with you. So here you will, if you want to do any kind of, services regarding molecular cloning, recombinant DNA, like DNA isolation, PCRs, microbiology. So these are the different, services that we provide, like the genomic DNA isolation services. So you just bring us a sample and we will isolate DNA from anything. So we just have, I think right now there's one person from Manipur University, he contacted us that he wanted to exotic bacteria that they have found. So these services we provide, we'll give you all the, you just have to bring a sample and we will do it for you. And then we do cDNA preparations, RNA isolation services, and whatever tissue samples or plant samples or any kind of samples that you have. We have electrophoresis services also for different RNA gels, DNA gels, urea page, and all those things. And then we provide all kinds of PCR services, from any small PCR fragment to other difficult templates which has high GC content, difficult to amplify. So you can just come to us, we'll get it done for you. And then we are actually maintaining a bacterial cell repository. So these are the bacteria. There are more to it. This list is a bit small. So you can just contact us, any bacteria isolate that you need. So we'll give you a slant bacterial slant, and that you can right away go to your laboratory and just keep it and put it for growth. Then we have custom molecular cloning services. If you want to clone your genes of interest, fragment of interest, we'll do it for you, and we'll just talk it over, and then we can get it done for you, right? So these are the services. This is just a view and pictures from my lab, people at work, and so these are contact details. For training programs, you can contact Reena. She will help you out. And for any other assistance, training, details, whatever, so Moshmi is always there anyways. So I think you can take the picture of this or whatever, I mean, right? And I think I'll end up here.
Thank you so much, Swapnil.
Yeah.
Dr. Sahil or Reena or anyone from BioAptagen?
Moshmi.
Moshmi,
Yes.
who would like to say something? Would like to
Sahil,
add
you want to Sahil, you want to add something?
Yeah, hi everybody. Am I audible?
Yeah, you're
yes,
audible.
yes.
So actually, whatever Swapnil has told, that is a very brief introduction. But I guess for the engineering part, so there is a science part that mostly Swapnil is developing in IIT, and then there is an engineering and the technology development where the detection happens. So I'll also discuss with Swapnil because detection is one of the most interesting phenomena of science where light has been used to detect various things. So if you get a chance to visit IIT, then there are lot of different equipment, lot of different labs that are there. So she was mentioning about nanoparticles. So my work is basically on nanoparticle dependent detection. So like when I was working in my bachelor's also, someday I went to see these equipments where at nanoscale, that is 10 raised to power minus 9, like if your hair is 1 micron, if you slice it in 1,000 part, then there is a nanoparticle. So if you see those nanoparticles from your nail, then you will get to know and you will develop a lot of interest, and then you will see like how the work at micro and nanoscale is there. So I guess these facilities are available in IIT, and Swapnil and our company is trying to actually give you that hint that science is an interesting field. Just theoretical knowledge is there, but then if you see atoms or these nanoparticles using these different electron microscope or FSM, then you will actually develop interest, and that is what I guess the purpose of the whole workshop would be. Like there will be certain techniques that we will be giving you, training we will be giving you, but we will try to actually give you the confidence that you can pursue science as your career. So that is what is the notion about all these activities. So
Right.
that's from my end, and there are like any specific question is there, you can write to me and you can ask now also, because once the science is done, then we are trying to convert those things into a product which can be used in the market.
Right.
So that's it from my end. Yeah. Yeah.
Thank you so much, Sahil. Anyone else from BioAptagen?
So we have Reena with us and we have Moshmi. So these are the two people that we have. If they want to say something, now is the time.
Also, the students have, participants have not yet posted any queries in the chat box. So maybe
Yeah,
we
okay.
can request the participants who have queries to, you can unmute yourself and ask your question also. Also, faculty members are there in the participants. So please feel free to ask or ask your queries to the BioAptagen team. Maybe I can start with my doubt about, or my question about
Sure.
how did it come about, the idea of combining the the aptamers with nanotechnology? What was the point of contact, you can say?
Like I said, no, we actually were trying before I went into, for nanotechnology and all. What I thought, we were actually preparing these TNA aptamers, we were thinking to conjugate it with some kind of an enzyme, the way ELISA works, right? But then we found that it was pretty difficult to do and the sensitivity was low, and that is how actually I came in I read papers and then I thought, why not do it in develop into a form of nano sensor. They are cost effective and they are sensitive also. So I think Sahil might speak much in that, how these nano sensors, they are have a plus point than other detection techniques. I
Yeah,
think he's the
particularly
best
if
person
they,
to.
yeah, particularly if they make it more affordable.
Yeah,
The
affordability is
diagnostics.
one point, and I think second point is also to make it more precise and
Right.
mess free. Because when you do ELISA, you need all those washing step, combining step, binding step, and lots of washing steps, and they just make your protocol a long protocol, right?
Right.
And that could be done only in the diagnostic center. So we just wanted to make something which is a point of care, and you don't need those many steps for getting the result, right? And why after sensors or kind of a nano sensors, they are better than other detection device? I think Sahil is the person to answer that question. Sahil, you are there?
Yeah.
Network issues are there in his side.
Yeah, I think there's some.
But if someone has any query, you can please go ahead. Sister Shiny, I think, has some query.
Yeah. I have a query that is regarding sending the students for an internship.
Okay.
Not only internship, if we want to send some students with particular work that designed by the students.
Okay.
So
Yeah.
what will be the criteria to you people will help them, or they have to go about their own way?
So what I mean, if they want to work on their projects, that's what you're saying.
Their projects, yeah.
Yeah, they can come here. We will help them out, and so we will set, what, because then we will be charging on the basis of what consumables that or techniques or instruments that we are investing in that. So in that case, it will become more of like a contract research kind of thing. We have both choices. They can come and work on their own. In that way, they will learn how to do the experiments. So we will let them use our infrastructure and our consumables, right? And so it's kind of a contract service, but then they will be working. So it's their choice. If they want to work on their own, they can do that, or they can just tell us that they want to get this thing done. So we can do that for them also. So both way it could work. So in that case, we don't claim on any of the patents or publications that you guys are doing, because that is not our work. So it's based totally completely on services basis, right? So it works that way.
Okay.
And for
Only that,
internship, they can come anytime if they want. We have different modules, and we are very flexible also. Say if some student, if they say, we don't want to learn this, we want to learn that, so we can just mix and match and make a specific model for them also. So we are specific in that. I mean, very flexible in that way.
so every kind of work would be possible to do in the lab, I feel, because many of the different types of equipments are there, I think.
Yeah, so
Molecular
we have
biology.
all the we have the full molecular biology lab. We have full cell biology lab, or the mammalian tissue culture lab. We have a full microbiology lab. Plus, we have this all the bioinformatics thing that we do, and all kind of protein methods. And we are actually trying to incorporate our training programs with IIT itself.
Okay.
That is still we
Yeah.
are talking through it, so that is why I didn't say that during the session. So we are trying to make it like with the IIT, not with the
Yeah.
faculties, but we are making it through IIT umbrella. So
Okay.
in that case, the plus point is that students will get exposed to all the IT instrumentations that they have. So there'll be a better exposures to different kind of instrumentation facilities and different techniques also. Like Sahil is an engineer, so he has different types of other modules. So
Yeah.
that will become the arena will become big, right?
Yeah.
So, but we have all functional labs with microbiology, cell biology, molecular biology, everything we have.
Okay. Okay, thank you. Yes, Priya.
Yes, if there are any further queries.
Hello, sir.
Yes.
Yeah.
Yeah. Prima.
Yes,
So I have this is Seema. Hello,
Yeah.
Swapnil, sir.
Yes.
Yeah. Hello, Sister Shiny.
Yes, yes, yes.
Hello.
Continue.
Yes.
Yeah,
Yeah,
yeah.
yeah,
Please,
can
go
hear
ahead.
you.
Yeah.
Hello, Swapnil. Thank you so much for the brilliant presentation. It was actually an eye-opening for me because I'm in
Thank
Pune
you.
and I'm a PhD scholar from Assam, Bosco, and
Okay.
ma'am, have said like your lab is like more into like guiding students for how to from the basic research to journey from the basic science research to translational applications and all. So my
Right,
first
right,
question was
right.
like when is there any time like when can like I'm looking for such guidance like
Mm-hmm.
from my whatever research I'm doing in my PhD. I would actually would like to take it a bit more further into a translational application. So
Mm-hmm.
developing states and all. It's a very eye-opening for me that such a big platform is in Guwahati also. And was one of my
Yeah.
question, and how you do the cardiac malfunction kit, how the work on cardiac malfunction actually works, because you said that the DNA aptamer based kit development is something what undertaking, and
Right.
also mentioned that in your current topic you also work on cloning of the cardiac
Mm-hmm.
biomarkers and all. So my one of my question is like if I know that responsible for causing a certain disease, how to take from that genetic level to that translational part or developing a diagnostic kit. Just you
Right.
insight onto that
Yeah, I mean,
journey.
if you want to do that, I mean, I don't know what research you are doing right now. So, but we can definitely help you in that. We can have you can work in our lab also. You can have some. We, I don't know what is your background. Also, what you have learned before joining your PhD. So, you can just give me a call anytime, and we can talk it over. At what is your requirements and what you want. How you are going to plan your research. We can definitely, so if you have a biomarker, what we generally do is if that depends in what kind of sensor you want to make for that and how feasible is to make that sensor, if that biomarker is present in your blood, if that biomarker is in your cerebrospinal fluid or in your urine or in your swab sample or in your a saliva sample, or if it is even if it is a cancer biomarker. So we are actually planning a next level of maybe after a year or so, we are planning liquid biopsies in which you don't have to do actually tissue biopsies, right? They are highly invasive procedures. So we want to check some kind of microRNAs that are present in your blood, like which are biomarkers for many cancers, prostate cancers, lung cancers. So these are present in your blood. So we are doing some kind of very initial R&D on that also. So it totally depends what kind of biomarker you're, working on, and then we can see how we are going to take it from developing that biomarker, converting that into a protein, then developing aptamers. Or if aptamer is a choice, we can go for any other sort of detection methods, DNA detection methods. So we can work it out. Just you need to talk to us. Just so you can talk to Moshmi, and she might set up a conference call. Then we can talk through what are your ideas, and then we can help you out definitely.
Sure, ma'am. Thank you so much. I got the contact details. I'll surely
Yeah, yeah,
contact
yeah.
you.
Yeah, sure, do that.
Yeah.
Okay, if there are any other queries? Okay, if no further queries, then I would like to thank Dr. Swapnil immensely for
Thank you.
firstly agreeing to be available today and to be there along with her team members. And it was a very expected and anticipated webinar. So, and it has lived up to that expectation. It has been really wonderful. And I found that apart from the innovative research and product development on APTA sensors, you have been able to find out a very important pain point in Northeast India, particularly with regard to training youth in biosciences or sciences as such, and the interdisciplinary fields in which we are looking ahead. So, like your suggestion was there to have students of engineering and other fields, because we also have departments of physics, chemistry, and other basic sciences here in Patricia, as well as the engineering students in engineering faculties in Azara campus. So we will try to organize a webinar on the same way for so that the engineering and other students also can be benefited from this very excellent venture of yours.
Thank you.
So let me take this opportunity to thank you and your team members, Dr. Sahil, Tarina, and Mosumi, for organizing and taking the pain to be available. I also wholeheartedly thank the participants who have joined in such large number and have. I'm sure they must have benefited by this presentation, by the webinar, and to the faculty members of our department. I am especially thankful for having contributed in organizing the event so properly in so little time. So I once again take the opportunity to thank you, Swapnil, for agreeing to be available today and making such a beautiful presentation.
Thanks for having me, and thank you all the students. Right, so we look forward to seeing you soon in our labs.
Exactly. That's what. Yeah.
Yeah, of course, we will send the students.
So the participants, kindly, I will be sharing a feedback link. So kindly send your feedback to the feedback link, and we will be issuing e-certificates to all the participants of the webinar.
Yeah.
So feedback link. So we would then wind up today. If Sister Shiny has anything to say?
No, once again, thank you. And it was wonderful talk. And the students are not asking questions because I feel this is little more higher research. Higher research, yeah. Higher research, and to understand also may find difficulty.
They can visit, even if they join or not join, that is a different actually outlook, but they are welcome to visit our lab, just walk around.
Yeah, it's actually, it's
so
actually, as Lima told, that it is an eye-opening webinar session. So we also will have that collaboration with you, your lab, and we have dissertation, and we have students interested in internship. If we have this facility in Guwahati, is wonderful.
Yeah, that's what.
Yeah.
See, that is what that Supriyo pointed out, the pain point.
Yeah.
There is no such thing in Northeast.
Northeast.
So that is why, because I have one of the anthropology students, she came, this winter, and she said, whatever I'm learning here, we used to go to Chandigarh and Delhi to learn.
Yeah,
And
yeah,
it's
yeah.
very good that we are having these facilities here. Right,
Yeah.
so that is, and she's a student of Guwahati University, and her HOD then contacted me and she said, it's very nice that you're having this, so we will send our students to your facilities. So that is
Yeah.
what we wanted, because I interact with a lot of Northeast students and I see a lot of potential here in students.
Yeah.
And the only thing they lack is that, state-of-art infrastructure. So that is what we thought, I just I really want students to gain whatever I have learned. And Supriya also, I mean, he knows many things. So that is what, we want to do it jointly with you. And that would be really great actually for the students.
And another facility is we have to send the sample for some analysis, even Bangalore or even Delhi.
That's what.
That is another facility.
Right. You don't have, it just adds cost and all those things. So you can just send it over to us. That is that is the idea behind starting Original. Because we want to do it everything in Guwahati. So we just want to, progress it. We are actually planning some more instruments coming in so that we can do a lot of other sophisticated techniques in the lab. So anytime.
Yeah, we can. One more. I am interested in. We can have one day workshop in a collaborative manner. It will give more audience.
Right.
It will be giving more audience, and that is this is virtual mode, but in a real.
Now we can do that. We can definitely plan with Sahil also. We can have an interdisciplinary kind of workshop of 2 days in collaboration with Bosco. So we can definitely plan it out. That will be a great idea. Actually.
So thank you so much, and very good talk and contribution to the Northeast. That is also very encourageable and a very good contribution. Thank you so much. We will meet in future.
Sure. I'm looking forward to it. Thank you.
Thank you, Sister Shaini, for your.
Okay, thank you, Supriya.
So then we'll close this seminar today. Thank you everyone for your active participation, and we will look forward to have such exciting seminars ahead, webinars ahead. Thank you again, Swapnil. Goodbye, everyone.
Goodbye.